All right, so today we're going to talk about metabolism. Metabolism is a really big topic, and we have just a short section here that we're going to be covering it. Metabolism is all of the chemical reactions that go on in an organism. We're not going to look at all of them. What we're going to be looking at is the stuff that is pertinent as to how your body gets energy and then how it will spend that energy. We're not going to look at all of the reactions of how you spend energy. That would be just way too many. So basically, what is going to happen to the nutrients you take in? Metabolism looks at the transformation of matter and energy. It is subject to the laws of thermodynamics. And looking at this, we break it down into metabolic pathways. So we'll start with a specific molecule. We'll look at the steps in order, how it is modified to form a product. One thing to keep in mind, each step is going to be catalyzed by a specific enzyme, but we're not going to be going into that kind of detail. The catabolic pathways are going to be the ones that break things down. In general, they release energy. The anabolic pathways are going to consume energy and they're going to build things. With cellular respiration, what we take is glucose, which is the C6H12O6, and it's going to react with oxygen, or in the presence of oxygen, to make carbon dioxide and water. The glucose you would get from consuming carbohydrates. Even if it's not directly glucose, most things are going to join into the pathway that glucose uses close to the top. If you consume things such as lipids or proteins, they actually will join into this pathway as well, just not at the top. Oxygen, we live in the presence of oxygen to be able to break things down. The carbon dioxide you're going to exhale, and then the water will be produced in your cells and used by your body. Bioenergetics is the study of how energy flows through a living organism. One of the key molecules we look at here is ATP. It's adenosine triphosphate. It's got a ribose, an adenine, and the 3 phosphate groups. So here is a little bit more detailed picture of it. These 3 phosphate groups are going to hold a lot of energy in the bonds. So the bonds of the phosphate groups get broken off by hydrolysis. And after you do that, here's water being added in, that's hydrolysis. It's going to become ADP, adenosine diphosphate, and then that phosphate's released. So this is going to release 7.3 kilocalories per mole of energy under standard conditions. So the delta G is just talking about the amount of work that's available. When you see a negative number like that with delta G, it means it's a spontaneous reaction. So for our cells, they're not actually standard conditions. It's a little bit different conditions inside the cell. It's actually going to give you 13 kilocalories per mole. So this reaction is 78% greater under cellular conditions. So types of reactions that we have, one of them is fermentation. With fermentation, you partially degrade the sugars. This occurs without the use of oxygen. Some of our cells can do this, some of them cannot. With aerobic respiration, oxygen is consumed as one of the reactants with the organic fuel. So your organic fuel would be the glucose. So the cellular respiration is technically going to include both some aerobic and anaerobic processes. So you have glucose reacting with the oxygen. Overall, it is aerobic. It's going to give you carbon dioxide, water, and energy. Energy is in the form of ATP and heat. The amount of energy we get from this is 686 kilocalories per mole. So our catabolic pathways themselves do not directly perform cellular work. They're going to use ATP like a chemical drive shaft. You can think of it like your paycheck. You go to work and get your paycheck, you use your paycheck to pay your bills. And it's a continual process. Cells are going to break down glucose to get energy as ATP, and then it will use that ATP to fuel other things in the cell. So redox reactions, this is a shortened term for oxidation and reduction. These are reactions that involve transferring electrons. So oxidation is the loss of an electron from one substance to another. Reduction is the addition of electrons to another substance. So you have a reducing agent that's your electron donor, your oxidizing agent that's the electron acceptor. We care about this because electrons are going to lose some of their potential energy along the way, and the energy gets released as they're being moved. So in metabolism, we are going to have some reactions that take place that are redox reactions where electrons are going to be moved, and they're going to release some of their energy as they do that. So the energy state of the electron changes as the hydrogen with its electrons is transferred. This is just showing here some redox reactions in here. So methane would be your reducing agent. So it's going to become oxidized. Oxygen is your oxidizing agent. It will become reduced. So our cellular respiration stages, we start out with glycolysis. This is going to occur in the cytosol. During this process, glucose gets split into two pyruvates. The citric acid cycle, also known as the Krebs cycle, also known as the TCA cycle, which is tricarboxylic acid cycle, occurs in the mitochondria. This involves the complete breakdown of glucose. It's going to oxidize derivatives of pyruvate into carbon dioxide. So in some of the steps, the electrons are going to be transferred to NAD+ to make NADH. That's one example. We will actually also have them transfer to FAD and make FADH2. Your electron transport chain is going to do your oxidative phosphorylation. So electrons are going to be passed from one molecule to another, and we call it an electron transport chain because this occurs at a chain of carrier proteins. So at one end of the chain, the electrons are going to be combined with the oxygen and the hydrogen ions to form water. The energy is going to be released and then stored to make ATP through oxidative phosphorylation. This is a nice overall picture of metabolism showing where the things occur. So in glycolysis, you split glucose in half to make pyruvate. So you're going to have some substrate level phosphorylation here. Basically, your phosphate's going to come from another organic molecule, and you're going to have some electrons be released onto NADH and carried that way. The pyruvate is going to get transferred into the mitochondria and become acetyl-CoA. This is going to enter into your citric acid cycle. The citric acid cycle will produce a little bit of ATP through substrate level phosphorylation, but it's going to produce a lot of NADH and FADH2. And these, in turn, are going to end up going into your electron transport chain, where oxidative phosphorylation will occur and chemiosmosis. That will generate a large amount of ATP at the end. So we're going to break down each of these cycles into more detail. So the term substrate level and oxidative phosphorylation. With substrate level phosphorylation, this is going to involve the phosphate coming from an organic substrate. So there's a small amount of ATP formed in glycolysis in the citric acid cycle this way, when an enzyme is going to transfer the phosphate group from an organic substrate. With oxidative phosphorylation, it's going to come from an inorganic source. So for each glucose that's going to get oxidized to carbon dioxide in water by respiration, we're going to make about 38 ATP. It's a net gain of 36 ATP with 73 kilocalories per mole of free energy formed. So we say a net gain of 36 ATP because we do have to invest a couple to get this process started. So these are the key reactions in glycolysis. So we start out with this glucose here. This is your energy investment phase because you're going to take 2 ATP and put them in. So these phosphate are going to be removed and you're going to have 2 ADP produced. Next, we have the energy payoff phase. You're going to get 4 ATP back out of this. You're also going to move some electrons and get 2 NADHs out of this. So it's an energy payoff. Your net at the end, your glucose, is going to be split in half to make 2 pyruvates and 2 waters. We have 4 ATP formed. You've invested 2 to get started. We have a net gain of 2 ATP. We put in the 2 NAD pluses. We end up with 2 NADHs out at the end. So we're going to look at these in a little bit more detail. So in glycolysis here, we start with glucose. And here in yellow, it allows you to follow the path of the phosphates. You can see on here how many carbons are in the molecules. Each place you've got a corner. Unless there's something else in it like oxygen, it's a carbon. So we're going to take the phosphate off of ATP, leaving ADP behind, and put it on carbon number 6 of glucose. That's why we call it glucose 6-phosphate. This is going to rearrange itself a little bit, and now it's fructose 6-phosphate. We need to put a little bit more energy in. We're going to take another ATP and put that phosphate in here on carbon number 1. So now we have fructose 1,6-bisphosphate. This finally has enough energy into it that it's going to split in half. One of the things it makes is dihydroxyacetone phosphate. The other one is glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is the one that's going to continue on to the next step. But these exist in equilibrium. So if we're constantly pulling the glyceraldehyde on, all of this dihydroxyacetone phosphate will eventually convert into the glyceraldehyde 3-phosphate and move on to the next step. So here we're going to integrate some inorganic phosphate and move some electrons. So we're going to have 2 NAD pluses become 2 NADHs. At this point, you have 2 of everything. So back here, you split this in half, essentially made 2 glyceraldehyde 3-phosphates. So everything else down the cycle, there's 2 of them. So the next thing we will have is these phosphates incorporated on here, and it becomes 1,3-bisphosphoglycerate. We're finally going to have enough energy that we're going to get some payoff. We're going to pull this phosphate off of carbon number 1 and put it on ADP. We're going to do it on both of those molecules, getting 2 ATP back. At this point, we've paid back what we've initially invested, and we've got 2 of the 3-phosphoglycerates. It's going to rearrange itself a little bit to make 2-phosphoglycerate. That will rearrange to make phosphoenolpyruvate. And then we're going to pull this phosphate off again, put it on ADP to make ATP, and end up with pyruvate. So if we come back to this, you invested glucose at the top, got 2 pyruvate out at the end, a net gain of 2 ATP, and a net gain of 2 NADHs are your key things that came out of that reaction. So now on to the citric acid or Krebs cycle. This will complete the oxidation of the organic molecules. So the pyruvate now is going to enter into the mitochondria to complete this cycle. It's going to do this if molecular oxygen is present. So your TCA cycle or citric acid cycle is going to occur between the inner and outer membranes in the matrix of the mitochondria. The electron transport chain will occur a little bit deeper in the cristae or the folds of the inner membrane and the inner membrane space. So here we've got the pyruvate. It's got 3 carbons on it and a transport protein. When it transports in, we're going to pull off a carbon and 2 oxygens as carbon dioxide. This is going to be exhaled. And what we're left with is these 2 carbons here with the oxygen and 3 hydrogens. It's going to move some electrons again, so we're going to get some more NADH. Coenzyme A is going to add onto it, and it becomes acetyl-CoA. So we have 2 carbons here that are left from that 3-carbon pyruvate. You're going to make 2 of these, and then that will give you 4 of the 6 carbons from your original glucose are tied up in acetyl-CoA. 2 of them have become carbon dioxide to exhale. So we need to turn them all into carbon dioxide. That's 2 of the 6 turned into carbon dioxide right there. Here we've got our citric acid cycle, Krebs cycle, TCA cycle, where we're going to produce NADH, FADH2, carbon dioxide, and GTP. GTP is very similar to ATP. It's guanosine triphosphate. Here, guanosine triphosphate is made, and it's just going to pass the phosphate onto ADP to make ATP. So it's a quick little easy conversion here. So we're going to have our acetyl-CoA come in. It's going to bring these 2 carbons in and add them onto this oxaloacetate to make citrate. The citrate is going to rearrange itself to make isocitrate. Now we're going to pull off 1 of the carbons and 2 of the oxygens to produce carbon dioxide, and we're going to move some electrons onto NAD+ to make NADH. So now 2 more of those carbons are accounted for as 2 more carbon dioxides, because remember, 2 of everything coming through here. The isocitrate will rearrange itself to make alpha-ketoglutarate, and we're down to 5 carbons. We're going to pull 1 of those carbons off again to make more carbon dioxide. And in the process, it will make succinyl-CoA. It's also going to move electrons and make more NADH. So at this point, we have pulled off 6 carbon dioxides from breaking down the glucose. Now we just need to rearrange these molecules back up to here so we can continue to do this process. So that process of rearranging is also going to release some energy. We're going to incorporate the inorganic phosphate here to make GDP into GTP, and then that GTP will form ATP. The succinyl becomes succinate. Succinate's going to be converted into fumarate. So this time we're going to put the electrons on FAD. That will produce FADH2. The FADH2 is going to be another electron carrier similar to NADH. So the succinate turned into fumarate, fumarate rearranged to malate, and malate rearranged back to oxaloacetate, moving some electrons again and creating more NADH. So the key thing that comes in here is acetyl-CoA. What you're going to get out of this set of reactions is NADH, FADH2, carbon dioxide, and GTP. So the electron transport chain is this collection of molecules embedded in the inner membrane of the eukaryotic mitochondria. So this chain will exist in multiprotein complexes. They're numbered 1 through 4. They're tightly bound to prosthetic groups that are going to help with the catalytic function. So these carriers will alternate between being reduced and oxidized as the electrons are going to move down the chain. This is going to be how we get those NADHs and FADH2s that are holding electrons turned into ATP. So chemiosmosis is the energy stored in the form of a hydrogen gradient across the membrane. In just a minute, we're going to look at how we create this hydrogen gradient. The proton motive force is when the electron carriers deposit the electrons in the inner membrane space, it creates this hydrogen gradient, and it's got the capacity to do work. We're essentially creating a battery here by separating the protons and the electrons. We can get them to do work as a result of them being separated. So from glycolysis, your substrate level phosphorylation of 4 ATP gave you a net gain of 2 ATP. Each NADH we've made in the process is going to give us 3 ATP. Each FADH2 is going to give you a maximum of 2 ATP. Occasionally there is variations in these where some of the intermediates get used, but focus on these numbers to learn the cycle. So your citric acid cycle will have 2 ATP made by substrate level phosphorylation from the GTP. So this is the electron transport chain here. So what's going to happen is your NADH here is going to drop its electrons off. And I don't know why it's cut off a little bit in this picture, but it's going to drop them off with this first protein carrier. The electrons are going to get passed along the chain, but when it does, it's going to pump the hydrogen ion or proton over to this other side of the membrane. And each time it goes through one of these protein pumps, it's going to move a proton over to the other side of the membrane. So for NADH, it's going to pump 3 of them across. FADH2 is going to cut in line a little bit. It's going to miss this first opportunity to pump the hydrogen ions across. So as it passes it along, it's only going to pump 2 of them across. This is why we get 3 ATP from NADH and only 2 from FADH2. So at the end here, you have these protons and electrons separated. The ATP synthase enzyme is going to bring these protons back and use that energy to put a phosphate on ADP and make ATP. It's also going to have these hydrogen ions available here to combine with oxygen and make water. The ATP synthase enzyme is located in the inner membrane of the mitochondria. It's going to be what is going to make the ATP from ADP. So it's like an ion pump in reverse. So basically what's going to happen is as those protons are going to come in, it's going to rotate this internal rod and catalytic knob and catalyze, putting this phosphate back on the ADP to make ATP. So we use this proton motive force to drive other kinds of work. When we look at the complete oxidation of glucose under standard conditions, it's going to give you 686 kilocalories per mole. When you phosphorylate ADP to ATP under standard conditions, it's going to give you 7.3 kilocalories per mole. So if we look at the efficiency of respiration, you make 38 moles of ATP times 7.3 kilocalories per mole, and you divide that by 686 kilocalories per mole, we have about 40% efficiency. That means about 40% of the potential energy in the glucose ends up being transferred to ATP. This may seem like not very much, but it's actually really pretty good. I know myself, I would not want to have more than that. If you're more efficient, it just means you've got to eat less. So it's actually probably higher because we know that the amount of energy is lower under cellular conditions. So you go, well, what happened to the 60%? It's lost as heat. And to say it's lost isn't really true. We use that heat to maintain body temperature. So the more metabolism you're doing, say you're exercising, you're going to generate more heat. So some of it will dissipate as sweat, but this is why you don't have to live in a room that's 98.6 degrees. As you're doing this, you're generating heat that keeps you warm. Just to give you an idea, the most efficient automobile converts only 25%. So you probably feel a little bit worse than that when you think of going to the gas station and filling your car, that only 25% of that is going to actually moving your car. So fermentation, this is going to harvest chemical energy without the use of oxygen. There's also no electron transport chain. But what needs to be present is a sufficient supply of NAD+ to accept the electrons during glycolysis. It's kind of the limiting reagent. If you're making chocolate chip cookies and you run out of chocolate chips, it limits how many cookies you can make. If you run out of NAD+, it limits how much glycolysis you can do. So there are multiple types of fermentation. It consists of glycolysis and a reaction to regenerate the NAD+. So it can transfer the electrons from the NADH to pyruvate or its derivatives. In humans, what we're concerned about is lactic acid fermentation. There's other ways out there. We're not going to worry about them today. So the pyruvate will be reduced directly by NADH to form lactate. It doesn't release carbon dioxide. That's why we use this. If you were releasing carbon dioxide in your muscle tissue, it would damage it. They used to think that the lactic acid would cause muscle fatigue. We blamed it for the delayed onset muscle soreness. That's not it. We know that the lactic acid actually clears out pretty quickly. We think it may be due to changes in the potassium levels as well as microtrauma in the muscles. It's not to say that the lactic acid is going to feel good in your muscles. It will give you some discomfort in the immediate time frame, but that's not what's making your legs sore 2 days after leg day at the gym. So that lactate can be converted back to pyruvate by the liver cells. So here, if we look at lactic acid fermentation, we're going to ignore the picture on the left. You go ahead and do your glycolysis. You take your glucose, make your 2-pyruvate. You get 2 ATP out of it. You're going to get 2 NADHs. The pyruvate is going to be converted back to lactate. In doing that, you use the NADH to make the NAD+ that you need to keep the glycolysis going. So this is sometimes referred to as the Cori cycle. So when we compare both aerobic respiration and fermentation, they're both going to use glycolysis to oxidize the glucose with a net gain of 2 ATP. You don't get ATP any other way with fermentation. Lactic acid fermentation is basically to allow you to continue to do glycolysis. NAD+ is the oxidizing agent that is going to accept the electrons from the food during glycolysis. That's why you need to have enough of it. The difference is how they oxidize that NADH back to NAD+ to sustain glycolysis. So in fermentation, its final electron acceptor is an organic molecule. So in what we look at here, lactic acid. In aerobic respiration, your final electron acceptor is oxygen. That's where you drop those electrons off at the end. It's going to have much bigger ATP bonuses that go with aerobic respiration. Aerobic respiration is going to give you 19 times more ATP than fermentation. Fermentation, you get 2. Respiration, you get 38. This is a comparison here. Both of them start out with this glycolysis here in the cytosol. If oxygen's not present, you're going to do the fermentation to make your lactic acid. If oxygen is present, you're going to do the aerobic cellular respiration, where it will go into the mitochondria, make acetyl-CoA, goes into the citric acid cycle. So it will always choose the more efficient pathway if it can. So we obviously are going to consume things other than glucose. Glycolysis will accept a wide range of carbohydrates. So most of them are going to be hydrolyzed to make glucose or other intermediates that are really close to glucose to enter into that cycle. Glycogen is a storage form of carbohydrate. We store this in the liver and muscle. So glycogenesis is the formation of glycogen. You'll do this when there is excess nutrients, more than what you need to use immediately. You will store some of it. Glycogenolysis literally means lysis of glycogen here, breaks down the glycogen. So when the blood sugar drops during exercise or when you're without food, this would be one of the places that you will turn to get a little bit of that back. Insulin and glucagon are going to affect this. Insulin's going to promote the formation of glycogen. When you eat carbohydrates, you're going to release insulin. It's going to say, I'm well fed. Let's use this energy, store what's excess. Glucagon is telling the body that the glucose is almost gone. We have to break down some of that glycogen. Epinephrine is going to promote the breakdown of glycogen in response to stress. So under stress, you want to have more glucose available to be able to respond to the situation. Cortisol will also do this. Epinephrine is going to be the one that does it in the quick and immediate time frame. Gluconeogenesis is the conversion of pyruvate back to glucose. So this can occur in the liver when the blood glucose is low. It's important that we have adequate glucose in the bloodstream. So this is one of the ways that the body will try and do homeostasis with that. This picture here summarizes some of these pathways here. So what we've looked at with the carbohydrates. Here, as they're turned into their sugars, glucose, going to go through glycolysis. Some of the carbohydrates will make sugars other than glucose. They come in really close to the top. That pyruvate becomes acetyl-CoA, goes into the citric acid cycle, undergoes oxidative phosphorylation, your electron transport chain, to give you lots of energy. So other things we consume, fats, they're going to be in the form of a triglyceride. So we're going to break some of those off. We're going to take the glycerol. It's going to come into glycolysis. The fatty acids come in at acetyl-CoA. Some of them can come in other places, but some a little before, some a little after. Your proteins, we break those down into amino acids. This cycle is all about breaking down the carbons. So we've got to pull off this nitrogen waste. And then depending on which amino acid, some of them will enter in at pyruvate, some of them will enter in at acetyl-CoA, some will come in the citric acid cycle. So with lipid metabolism, your triglyceride is a glycerol molecule with 3 fatty acids attached. Lipolysis is just the breakdown of lipids. We do this through beta oxidation. So what we're going to do is break off 2 carbons at a time, and they will enter in at acetyl-CoA. So the fatty acids enter in at acetyl-CoA, the glycerol goes in at glycolysis. Fats are going to yield more ATP than glucose. When you look at a fatty acid having 16 to 18 carbons in there, when you break it off 2 at a time, that's a whole lot more acetyl-CoA that is being produced from a fat than is being produced from glucose. So it's going to take more turns of the cycle to break them down. Lipogenesis is fat formation. So this will occur with excess nutrients. So again, the body says this is more than I need to use immediately. Let's store some of it. So this is just showing here what a triglyceride looks like. You've got a glycerol molecule, and then these are your ester linkages, hooking these fatty acids on there. So looking at the lipoproteins or apoproteins, we've got 4 different categories of these. The chylomicrons, VLDLs are very low density lipoproteins, LDLs are low density lipoproteins, and then the HDLs are high density lipoproteins. So this is showing here where you've got your cholesterol and triglyceride inside the molecule with this coating around the outside. So your chylomicrons are going to form in the small intestine in the mucosal epithelial cells. They're going to enter into your lymphatic system and then eventually the blood. So the capillary surface enzymes are going to hydrolyze the triglycerides. So the fatty acids and triglycerol enter the fat cells, and then they can be resynthesized into triglycerides for storage. The chylomicron remnant is then degraded by the liver. So the VLDLs, these are produced by the liver. They're going to transport lipids to the adipose tissue for storage. So the triglycerides are removed, and then it's going to become an LDL that will contain mostly cholesterol. So the cell is in need of cholesterol for membrane repair, and steroid synthesis will absorb the LDLs by endocytosis. So the LDLs are actually used for a good thing for their cells, but the LDLs are a lot of times considered the bad cholesterol, because if you have a lot of LDL, it meant you did a lot of this drop off at the adipose tissue. It indicates more fat storage. So the LDLs carry 75% of the total cholesterol in the blood. They're going to deliver it to the body cells for repair and synthesis, but it can deposit cholesterol in fatty plaques. So high density lipoproteins, they're the good cholesterol. They're used to remove the excess cholesterol from the body cells and the blood, and they'll deliver it to the liver for elimination. So the liver is going to produce an empty collapsed protein shell. This can travel through the blood and pick up the cholesterol. So when it returns to the liver, it can be removed and eliminated. One of the ways we can eliminate it is bile or your bile acids. So we like to maintain a high level of HDL because it indicates cholesterol is being removed from the arteries. You want to have a low level of LDL because it signifies that the rate of cholesterol deposition is under control. If that gets too high, you're depositing too much. So smoking, high intake of saturated fats, coffee, and stress, among other things, will increase the LDLs. Most cholesterol is actually going to be synthesized in your body, but dietary restriction may lower blood cholesterol in some people. Some people, their bodies are going to synthesize too much, and they can consume zero of it. Their body will still produce too much. Vigorous exercise will help to lower the blood cholesterol. Lipoprotein lipase is an enzyme found primarily on the surface of cells that line your small blood vessels within the muscles and in your fatty tissues. This plays an important role in breaking down fat and forming triglycerides. Those are carried to the various organs by the molecules of lipoprotein. So this is a summary of some of what goes on here. We've talked about glycolysis, going through to produce your pyruvate, then acetyl-CoA goes into the Krebs cycle or TCA cycle. Your triglycerides, these will form glycerol and fatty acids that can come here. This is showing that it is a reversible arrow, you'll note here. So if you have excess of these things, you can turn them into stored triglycerides. So we can have ketones produced, which we will talk about later. We'll talk about the amino acids later. So with protein metabolism, your amino acids are going to either be oxidized to produce ATP, or we use them to synthesize new proteins. So when you consume protein, you'll break it down into the amino acids. You're going to use what you need to do building and repair. But if you consume more than what you need for building and repair, then we have to deal with those. So these excess dietary amino acids are not excreted. They'll be converted into glucose through gluconeogenesis if they can, or they can be converted into triglycerides doing lipogenesis. There's no storage of amino acids themselves. So sometimes people think, well, they'll just turn into muscle. They're not going to turn into muscle if there isn't strength training to tell the body to do that. If there's just too many amino acids, they'll turn into fat stores. So absorption in the body cells is stimulated by the IGFs and insulin. With protein catabolism, you're going to have protein from worn out cells that will be broken down into their amino acids. So in addition to the protein that you eat, you also get some that's going to be recycled from your worn out cells. So before entering into the Krebs cycle, the amino group must be removed. So we call this deamination. When we do, it produces ammonia, and then the liver cells will convert it to urea, and that gets excreted in the urine. So transamination is going to be a chemical reaction that's going to transfer an amino group to a keto acid to form a new amino acid. So this is showing some transamination occurring here in the liver. It's showing here your amino acid being broken down, producing a keto acid, and here producing the ammonia that will be made into urea. It travels through the bloodstream to the kidney. The kidney is going to filter it out and remove it in your urine. So ketosis occurs when the blood ketone levels are higher. Normally, blood ketone levels are really low. Some tissues can use ketones for ATP production. So during fasting, starving, or a high-fat meal with few carbohydrates, where you've done a lot of beta oxidation, you can have ketone production. You can also have it after prolonged exercise. So you'll see this sometimes with endurance athletes. Go volunteer at the end of a marathon and you will smell the ketones on people. So acidosis or ketoacidosis is having an abnormally low blood pH. So some people will describe it as a sweet smell. You can smell it on a person's breath. If they've been producing them quite a bit, you'll smell it coming in their sweat as well. So this occurs in diabetics because the triglycerides are used for ATP production instead of the glucose, and the insulin inhibits lipolysis. In a diabetic, this can be dangerous because they're not good at controlling their blood sugar. This is just another picture here that integrates these nutrient pathways. You've got glycolysis, showing where you can have those keto acids come in. Here at acetyl-CoA, this is where you would produce the ketone bodies. So the fatty acids coming in here, they can produce ketone bodies. Some of your keto acids coming in, when you have excesses here, will produce ketone bodies. This is showing here where you do the deamination to get rid of it. So this is kind of a summary showing what happens when you have glucose in the bloodstream. Some of it can be converted into glycogen stored in the liver and the muscles. In most of the tissues, you're going to do this oxidation. This is showing here what the hepatocytes do with the proteins, the fatty acids. Here you can do some triglyceride formation, some glycogen formation. And then this is showing how things can end up being stored in the adipose tissue. So that is the summary of metabolism. What I would recommend doing is studying this in smaller pathways. Study just glycolysis, just the Krebs cycle, just the electron transport chain alone, and try and let them sink in and make sense before trying to get the overall big picture of it.